Hello students, welcome back to my class. In the previous class, we are discussing few household products, that is role of microbes in household products. And in the last class only, I have discussed the role of microbes in the preparation of bread, in the preparation of idli, dosa, and how the microbes play an important role in the curdling of milk. So in all these cases, the role of microbes is very, very important, and name of the microbe is also very, very important. You should remember. So let us recall few points that we have studied in the previous class, because we are continuing with that class only. So in the curdling of milk process, I told you the main microbe involved in the curdling process is lactobacillus or lactic acid bacteria. So those bacteria, lactic acid bacteria, they are usually present in the curd. When you add a small amount of curd, that is a starter, to the milk, the lactic acid bacteria which produces certain acids, and those acids coagulates with the milk protein called as casein, so that it helps in conversion of milk into curd. And next, in the preparation of idli and dosa, the main microbe involved is either bacteria or fungi. If it is a bacteria, Streptococcus picalis, and commonly available is the Saccharomyces cerevisiae. So Saccharomyces cerevisiae, it is commonly available everywhere, and wherever the sugar content is available, there we can find the presence of yeast. So that helps in preparation, that is used in the preparation of dosa and idli. And even the same Saccharomyces cerevisiae, baker's yeast, it is also used in the preparation of bread. So everything we have discussed in the previous class. Now we will be discussing a few more household products, that is role of microbes in household products. In that, the first one is toddy. So toddy, it is very, very interesting. So toddy, it is one of the traditional drink in some parts of the South India, and which is also prepared by the fermentation process of the sap obtained from the palm trees. Sap. You may be heard in case of plant anatomy of plants. There, a sap can be obtained from the trees. Sap is nothing but it is like a liquid. The sap obtained from the trees. So that sap is collected from palm trees. So which are the palm trees? So palm trees are nothing but the coconut tree, date palm, etc. So various palm trees are available. So from those palm trees we can collect the sap. So that sap is only later called as toddy, and it is a refreshing drink. The toddy is called as a refreshing drink. Why? I will tell you later. So this is a traditional drink, a refreshing drink in India. So it is obtained from the fermentation process of the sap obtained from the palm tree. So especially the Indian traditional drink obtained from the palm tree, it is called as Caryota urens. So this is one of the palm tree in which the sap is collected. Later it is used as a toddy, that is a traditional drink or a refreshing drink. You may have seen in certain cinemas, or you may have seen real. So for some palm trees, the pot will be hanged to collect the sap. So later that sap is used as a toddy. So in local language we can say it is kallu. It is called as kallu or neera, it will be called. So this traditional drink, it is commonly used in southern part of the India. Then how it is obtained? As I said, so in the tree, the flower or inflorescence part will be cutted, so that the sap will be coming out. That sap will be collected into the pot, and later that collected sap, it will be allowed for fermentation process. So fermentation, it is the main mechanism involved in the preparation of all household products. Even in the preparation of toddy also, so it is allowed for fermentation. And during fermentation, which is the microbe involved? Here also the Saccharomyces cerevisiae, it is naturally occurring in the toddy that undergo fermentation. During fermentation, it will form a beverage, that beverage containing 6% alcohol. What the sap you collected, it is allowed for fermentation process. During fermentation, the Saccharomyces cerevisiae, that will form a beverage. That beverage is called toddy, and this toddy contains 6% alcohol. So this alcohol percentage is very, very important. Toddy contains 6% of alcohol, and as it contains alcoholic content, content, it is called as a refreshing drink. Then this toddy later it is heated. The toddy it is heated to produce palm sugar or palm jaggery. You may have seen, some village people may have seen, it is called as tati bella. So tati bella, it is nothing but palm sugar or palm jaggery that is mainly obtained from the toddy when it is subjected to heating. When it, how the sugarcane juice is heated to produce jaggery. Likewise, this toddy is heated to produce palm sugar or palm jaggery. That is also very important. So this is about the toddy. Toddy, it is a traditional drink and it is a refreshing drink collected from or obtained from the fermentation process of sap obtained from the palm trees. One particular example for palm tree is Caryota urens. And how it is obtained? So the sap collected, it is allowed for fermentation. During fermentation, which is the microbe involved? It is Saccharomyces cerevisiae, and that Saccharomyces cerevisiae during fermentation, it will form a beverage which contains 6% alcohol. Which contains 6% alcohol. So that is toddy. And another interesting thing we can know about this toddy is that so this toddy it will be consumed within 24 hours. So after collecting, for a few hours it is allowed for fermentation so that it gets converted into alcohol, a beverage containing 6% alcohol. Within 24 hours it has to be consumed. So after 2 or 3 days, the alcohol content which is present in the toddy, it gets converted into acetic acid so that this fermented toddy later used as vinegar. Now, what the vinegar you are using, it is nothing but a fermented toddy. So, within 24 hours it has to be consumed. If it is allowed for a few more days or few more weeks, the alcohol content gets converted into acetic acid, and that acetic acid is nothing but vinegar. So, in the vinegar, the main content is acetic acid, as here also acetic acid. The ethyl alcohol gets converted into acetic acid means it is used as vinegar. This is about toddy. And next one, we are discussing about cheese. So cheese also very important and interesting. Cheese is one of the oldest milk product obtained by the process of fermentation, and it is also very important. You should remember it is the oldest milk product obtained with the help of microorganisms. Then how this cheese is prepared? See, for example, you know that how the curd is prepared, you know that. Later in that curd you can observe a solidified part as well as the liquid part. The curd, the main curd is separated from the liquid milk. In that curd, you are separating the curd from the liquid part of the milk. Later, that curd, it will be allowed to subject for hanging, or either it will be added to lemon juice. If the lemon juice is added, the liquid part will be completely separated, and what the solidified content will be there, that will be used for the preparation of cheese. And depending upon the water content, the cheese may be called as a soft cheese. If it is soft, the water content is about 50 to 80%. If the cheese contains 50 to 80% of the water content, it is called soft cheese. If the cheese contains about 45% of the water content, it is called semi-hard cheese. If it contains less than 40% of the water content, it is called hard cheese. So, depending upon the water content in the cheese may be called as a soft cheese, semi-hard cheese and hard cheese. And you know that different varieties of cheeses are available, and those different varieties of cheeses are mainly based on depending upon the texture, flavor and color, even it is depending upon the taste. So there are different varieties of cheeses, their texture is different, flavor is different and even the taste also different. But main thing, the different variety of cheeses are that specificity what we are saying that mainly depends upon the particular microbe which is growing or which is involved in that preparation. You should remember the specificity or variety of cheeses are mainly depending upon the type of microbe used. For example, one particular cheese called as Swiss cheese. Swiss cheese. So this you may have seen. Real, or if you have seen Tom and Jerry, in that you can see like this. So this is a large cheese having large holes. So Swiss cheese is characterized by the presence of large holes, and what may be the reason for the production of these large holes? So this is the characteristic feature. So the large holes in Swiss cheese are due to the production of large amount of carbon dioxide by a specific bacteria called as Propionibacterium shermani. Propionibacterium shermani. It is the specific microbe or bacteria involved in the preparation of Swiss cheese. It is very important, most asked question in your NEET examination. So the large holes in Swiss cheese are due to the production of large amount of carbon dioxide by a bacteria called as Propionibacterium shermani. So this characteristic feature is obtained by the addition or with the growth of this microbe. And one more type of cheese is called Roquefort cheese. Roquefort cheese. So this Roquefort cheese, it has a specific color as well as specific flavor, and this specific flavor is obtained by the ripening of this Roquefort cheese with the help of a specific fungi called as Penicillium roqueforti. It is a fungi. You should remember it is a bacteria. So this Penicillium roqueforti, it is a fungi. So this fungi helps to produce a specific flavor and color to this Roquefort cheese. So Roquefort cheese, it will be ripened with the help of Penicillium roqueforti. And one more type of cheese is called Camembert cheese. Camembert cheese, and this Camembert cheese is obtained or ripened with the help of a growth of a fungi called as Penicillium camemberti. So these two are the fungi which are mainly involved in the production of a specific type of cheese. So preparation of cheese is very important. Cheese, it is one of the oldest milk product obtained by the fermentation with the help of microorganisms, and the curd is separated from the liquid part to obtain the cheese. Depending upon the water content, different varieties of cheeses are available, and the variety or specificity of the cheese mainly depending upon the type of microbe involved. So in the Swiss cheese, the large holes in Swiss cheese are due to the production of large amount of carbon dioxide which are produced by a specific bacteria, Propionibacterium shermani. And the Roquefort cheese, so that Roquefort cheese, it has a specific color as well as a flavor because of the Penicillium roqueforti. And Camembert cheese, it is ripened with the help of Penicillium camemberti. So these are certain different types of cheeses which are mainly depending upon the type of microbe involved. And even the microbes are used in the fermentation process of fish, soya, and even to ferment the bamboo shoots. You may have seen in other countries the bamboo shoots are used for the edible purpose. So before eating, they will be subjected to fermentation process. So what I am saying, the microbes are even involved or used in the fermentation process of fish, soya, and bamboo shoots. So which is the microbe involved in the preparation of soya sauce? You may have heard about the soya sauce, different type of sauces. So in the preparation of soya sauce, the main microbe involved is Aspergillus oryzae. Aspergillus oryzae, it is the microbe involved in the preparation of soya sauce. And another one, fish sauce. So this fish sauce is obtained from different microbes like Streptococcus, Micrococcus, Bacillus, etc. are involved in the preparation of fish sauce. And even in the fermentation process of bamboo shoots, the lactic acid bacteria mainly involved in the fermentation process of bamboo shoots. So these are certain household products where the microbes play an important role. So all these products, all these microorganisms are very important. So this completes the role of microbes in household products. So if you have any doubts, you can ask me later. I can able to explain in the next class. So now we will discuss a few objective questions related to the microbes in household products. So I will send them these objective questions in your group. You can see. So now let me discuss the objective questions related to this topic. So the first question, where do we find microbes? The options are like this: deep in the soil, option B in thermal vents, option C under layers of snow, and option D all of these. You know that I told you microbes found everywhere, even in the places where no other life forms exist. So here when you see all the options, all options are correct. In all the options we can find the presence of microbes. And second question, which of the following microbe is a proteinaceous infectious agent? Option A, viruses. Option B, prions. Option C, viroids. Option D, protozoa. So here when you discuss about the viruses have specific characteristic features. Prions also, viroids also. So here the proteinaceous infectious agents are nothing but prions. So all these you have studied in your biological classification chapter. So here prions are the proteinaceous infectious agents. And next question, the microorganism used in curd formation. Option A, Aspergillus. Option B, Saccharomyces. Option C, Lactobacillus. Option D, Penicillium. So you know that in the mainly during the curd formation, Lactobacillus or lactic acid bacteria are involved. So option C, Lactobacillus is correct. Then the fermenting agent of dosa and idli is. So during fermentation of dosa and idli, which is the main microbe involved, let us see the options. Saccharomyces cerevisiae. Option B, Leuconostoc mesenteroids. Option C, Streptococcus faecalis. Option D, both A and C. You know that the bacteria Streptococcus faecalis as well as Saccharomyces cerevisiae, Baker's yeast, both are involved in the fermentation of dosa and idli. So your option D. And fifth question, leavening in bread preparation is caused by the action of enzymes. So leavening, I told the swelling up of the dough, that is called leavening, and that leavening in bread preparation is mainly caused by the action of three enzymes which are produced by the Saccharomyces cerevisiae. Then which are the three enzymes? First we'll see the options. Option A, amylase. Option B, maltase. Option C, zymase, and option D, all of these. So here all the three enzymes are involved in the leavening process of the bread. Next question. The soft and porous nature of bread is due to. So why the bread becomes very soft and porous? Option A, due to the production of carbon dioxide only. Option B, ethyl alcohol. Option C, both A and B. Option D, none of these. I told you during fermentation process both carbon dioxide as well as ethyl alcohol are liberated. So when the dough, leavened dough is subjected to baking, the carbon dioxide and ethyl alcohol will be evaporated and makes the bread soft and porous. So your option both A and B. The next question, toddy is a. Option A, traditional drink. Option B, refreshing drink. Option C, obtained from sap of palms. And option D, all of these. So here all are correct. I told you, toddy, it is a traditional drink, a refreshing drink, and mainly obtained from the sap of the palms. So these are certain objective questions that I have discussed in this class, and few more questions that will be given home assignment to you. You can able to find out the answer. If you are not knowing, let me know that I can discuss in the next class.